Hello everyone and welcome to Agri Talk Science. I'm Tony and today we are diving into one of the most exciting and rapidly evolving areas in agriculture, omics technologies. These cutting-edge approaches are transforming how we improve crops, especially as we face growing pressure from climate change and rising global demand.
Hey folks, I'm Zinga. So now let's face it, our food systems are under serious strain because by the year of 2050, we could have over 9 billion mouths to feed. And that challenge is being made even tougher by extreme weather, prolonged droughts, record-breaking heat waves, and ever-evolving pests and diseases. Crops are under siege and farmers need better tools fast.
Exactly. Traditional crop breeding, while still essential, just isn't fast or precise enough to meet today's complex agricultural challenges. So, what's the solution? Interfunctional genomics and proteomics, not just theoretical concepts, but powerful tools that let us pinpoint the exact genes and proteins responsible for key traits like drought resistance, yield, and nutritional value. Think of it as combining a genetic blueprint with a real-time performance report, giving scientists and breeders a clearer, faster path to building better crops.
And that's what we are unpacking in today's episode. How these technologies are being used in real-world agriculture to build crops that are more resilient to climate shocks, more nutritious for consumers, and more efficient for farmers. Plus, later in the show, we're bringing in our friends
Chian and Amirul,
who'll give us inside scoop on the proteomics side of the story, how proteins reveal what genes alone can't.
Let's break it down. Functional genomics is a powerful approach that helps scientists uncover the links between specific genes and key agronomic traits like drought resistance, yield potential, or nutrient content. Instead of relying solely on trial and error breeding over multiple seasons, we now have advanced tools like genome-wide association study (GWAS) and marker-assisted selection (MAS). This allows researchers to identify valuable genetic markers early in the breeding process, making it possible to predict plant performance and accelerate the development of better varieties.
Exactly, and the real-world impact is huge. Take the GS3 gene in rice. It plays a major role in determining grain size, which is a critical yield factor, or the GN1A gene, which boosts the number of grains per plant. So, by identifying and targeting these genes, breeders can create rice varieties that are not only more productive, but also tailored to specific growing environments. These genetic insights are helping to push the boundaries of what's possible in crop development with unprecedented accuracy.
And that's vital, especially as the climate becomes increasingly unpredictable. Genes like DLEB, dehydration responsive element binding, and HKT1, high affinity potassium transporter 1, have emerged as heroes in the fight against abiotic stress. And DLEB regulates how plants respond to drought, activating protective mechanism when the water is scarce. Meanwhile, HKT1 helps manage salt stress by controlling sodium ion transport, a key factor in maintaining cellular balance under saline conditions. These genes are literally helping plants survive where they once couldn't.
Absolutely, and beyond survival, let's talk about nutrition. So, functional genomics is enabling biofortification. So what is biofortification? Let me explain it briefly. It is a process of enriching crops with essential nutrients. With gene editing tools like CRISPR-Cas9, scientists have developed crops like golden rice, which contain enhanced levels of beta-carotene, a precursor of vitamin A. That's a huge win in fighting vitamin A deficiency in many parts of the world. We have also seen increases in iron and zinc content in staple grains like wheat, maize, and millet, helping to combat hidden hunger in vulnerable populations. So, ladies and gentlemen, it's not just about how much food we grow, but also how much nutrient dense it is.
That's a great point, but we can't forget the constant threats from disease and pests, which wipe out huge portions of global harvest every year. Genomics has given us a leg up here too. Yes, scientists have identified resistant genes such as NBS-LRR genes that recognize pathogen attacks and trigger immune responses in plants. By either breeding these genes in or editing key regulators like the MLO genes, we are giving crops a sort of genetic shell, helping them resist fungal disease like powdery mildew.
And if you've heard of BT crops, you've already seen genomics in action. Yay! So, these are genetically engineered to produce insecticidal proteins derived from bacterium called Bacillus thuringiensis. So, this protein specifically targets pests like corn borer or bollworm without harming beneficial insects or humans. What's even more impressive, by stacking multiple CRY genes, different variations of the BT protein, we make it far more difficult for pests to develop resistance. It's like hitting them from several angles at once, keeping our defenses strong.
Thanks Guy, I'm Chen and now we are diving into proteomics, the large-scale study of proteins and how it reveals what plants actually do when they are stressed. Unlike genomics, which show potential, proteomics capture real-time biological responses, helping us understand which proteins are activated under specific environmental stressors.
Hi everyone, I am Amirul. Proteins are the molecular effectors in plant stress responses, directly influencing cellular functions and metabolic processes. And unlike the gene expression at the transcriptional level, which may not directly correlate with the protein abundance, so proteomics allow us to examine the proteins that are actively involved in the stress adaptation and survival. So when plants encounter such environmental stresses, they will undergo a significant proteome reprogramming. So proteins involved in stress signaling, antioxidant defense, cell wall remodeling, and such metabolic adjustment, which are modulated to help the plant cope with this adverse condition somehow.
For example, during cold or low temperature stress, plants upregulate GAPTH, glyceraldehyde 3-phosphate dehydrogenase, and PGK, phosphoglycerate kinase, to support glycolysis. This action keeps energy production going despite reduced photosynthetic activity. This also ensures that the plant survives during energy-limited conditions.
So when it comes to the salinity stress, the major issue is oxidative damage from the reactive oxygen species or ROS. In response, the plant activates ascorbate peroxidase, APX, and glutathione as transferase the GST to detoxify the ROS and protect cells from the oxidative stress. So now for the heat stress, plants will rely on a group of chaperone proteins which called as heat shock proteins, HSPs, including the 70, 90, and SHSP, the small heat shock proteins. This will eventually prevent the protein aggregation and assist in a proper folding of the stress-damaged proteins, helping the plant recovering faster from the high temperature.
To study the dynamic change of protein in plants, Researchers use tools like 2D gel electrophoresis to separate proteins by their isoelectric point and molecular weight. Meanwhile, techniques like mass spectrometry was used to identify them with high precision data such as molecular mass and sequence. This reveals a detailed proteomic profile under each stress condition.
And by comparing these two profiles, we can identify the stress-specific biomarker, which eventually indicate how well a plant tolerates drought, heat, or salinity. This helps in selecting or engineering crops that express beneficial proteins at the right time and in the right moment.
So, proteomics is not just a research tool. It's a gateway to developing stress-resilient crops that can adapt to climate change. It gives us the clearest picture yet of how plants fight back on a molecular level. Alright, now let's shift our focus from plant stress to what ends up on our plates, because proteomics does not stop at resilience. It plays a huge role in food quality, nutritional profiling, and safety assurance. By analyzing the entire protein content of a crop, we can uncover what's beneficial, what's risky, and what needs to be improved.
Yes, exactly. So with powerful tools like the LC-MS or liquid chromatography coupled with the tandem mass spectrometry, we are able to identify and quantify the proteins present in the food. So even if in a very small amounts, this kind of precision helps the breeders and food scientists to enhance the nutritional values, monitor the processing effects, and flag such potential hazards long before the product reaches consumer.
Now, take high lysine maize for example. Lysine is an essential amino acid that our body can produce on their own. It's especially important in regions where diets rely heavily on cereals. Proteomic analysis confirmed that this biofortified maize contains higher level of lysine-rich storage protein, improving its nutritional impact significantly without introducing any unwanted or harmful proteins.
So, when it comes to allergen detection, proteomics is incredibly valuable. Many food allergens like the peanut protein ARHH1, H2, and H3 can actually remain active even after the food is processed or cooked. Traditional tests might miss them, but the proteomics can detect them in trace amounts, giving manufacturers a way to accurately label the food and protect people with severe allergies.
That's a big win for consumers' trust and safety. And it does not stop there. Proteomics also helps identify and remove anti-nutritional protein. These are compounds in some crops that blocks nutrient absorption or interfere with digestion. By minimizing this protein through breeding, we can make food not just healthier but also more bioavailable, meaning that your body can actually use the nutrients more effectively.
It's also incredibly useful for food spoilage detection. So a good example is in fish. As the freshness declines, certain muscle proteins like the troponin T will begin to break down. These fragments will act as a spoilage biomarkers, and proteomics can detect them early, long before the fish start to smell or look off.
And then, there's the role of proteomics in product authentication and fraud detection. It helps ensure that what's on the label matches what's inside the package, especially important for products like milk, meat, or plant-based food, where substitution or additive might be used.
And in short, proteomics gives us a deep insight into what's really in our food, from boosting key nutrients to flagging hidden allergens to ensuring the freshness and transparency. It helps us build a safer, smarter, and more nutritious food system.
It's all about moving from guesswork to precision agriculture and food design. With proteomics, we are not just feeding the world, we are doing it more responsibly from genes to plates.
So, if you think of genomics as a blueprint, it's like a detailed architectural plan. It shows us what genes could potentially do under ideal conditions, laying out the possibilities for crop traits like yield, resilience, and nutrition.
Then, proteomics is the real world inspection report. It tells us what those genes are actually doing inside the plant, especially under stressful environments like drought, heat, or poor soil. When we combine what the blueprint says with what's actually happening on the ground, we get an incredibly powerful toolkit for making smarter breeding decisions.
Exactly. This integrated view is giving researcher and breeder the upper hand. We can now respond to problems like climate stress, nutritional shortfall, and also the emerging pests more effectively and in record time. Instead of relying on years of field trial, we can shorten breeding cycles using real-time biological data, moving beyond just physical observation to molecular level insights.
And let us remember, the goal isn't just advancing the science for its own sake. At the heart of all this is a very real need: feeding a rapidly growing global population. We are doing this by using smarter methods that help preserve our natural resources, water, soil, and biodiversity. It's all about producing more with less and doing it in a way that's truly sustainable for the planet.
And what's ahead is even more promising. With the power of AI, machine learning, and precision agriculture, we are entering an era where we can model how genes, proteins, and environments interact. That means we will soon be able to make hyper-targeted agricultural decisions rooted in data and tailored to specific conditions.
So, ladies and gentlemen, just imagine it. A farmer able to assess real-time omics insight, weather forecasts, and soil profiles through a smart dashboard, and then choosing the perfect seed for that season and location. That's not next-gen farming. That's intelligent, adaptive agriculture already taking shape.
And this is not just a science fiction. This transformation is happening right now. Omics technology are already serving as the bridge between research lab and also the real-world field, turning cutting-edge breakthrough into practical, impactful farming solutions.
And with the ongoing innovation, collaboration, and the right global investment, we truly have the potential to build the food systems that are resilient, nutrient-rich, and sustainable. Thus, from gene to plate, ensuring the food security at all.
That's it for today's episode of Every Talk Science. Big thanks to Zinger, Jian, and Amir. Amazing insights all around.
And thank you all for tuning in. If you enjoyed this, share with your friends and fellow agri-geeks.
Until next time, keep growing smarter.
And let science lead the way.